Okay, so we missed the slide, right? We missed the previous one, but it wasn't much. So, okay. So now you need to understand it. And, all of these enzymes are not in your syllabus, but you still need to know them. So like I said, very easy stuff. You need to see that these two nucleic acids making a DNA eventually become four, right? And these four are just anti-parallel to each other. And then the other thing you guys know is that semi-conservative replication of DNA takes place. And all of these blobs and these labeled things are the enzymes. You can appreciate that the red one is the backbone. And I told you guys, what is the backbone made up of? I told you it was phosphodiester bond, which means it is made up of both the phosphate and the ribose. So appreciate over here that both the phosphate and the ribose, both structures are involved in this backbone. And what's involved in with hydrogen bonding is the base pairing. And hydrogen bonding is spontaneous. We've been doing hydrogen bonding since topic 1 when we did water. Hydrogen bonding is a spontaneous process because of distribution of charges, right? Distribution of electrons is a property of hydrogen to form hydrogen bond in whenever it's attached to, let's say, something like oxygen or nitrogen with heavy negative charges. So then, do you need an enzyme for hydrogen bonding? Not at all. You don't need one. But you need an enzyme definitely for this phosphate backbone or this phosphorus sugar bond. For that purpose, you have one enzyme which is known as the DNA polymerase. You can appreciate on both of these strands, DNA polymerase is working. These strands look fragmented, which means this one is definitely the continuous strand. This one is definitely the leading strand. And this one is the Okazaki fragments. These yellow and green ones are definitely the base pairings. This red one is definitely the backbone. Okay, and then we have DNA polymerase is working on them, working on both of these strands. Okay, the other thing you need to know is that, like I keep telling you, that DNA cannot be opened from one end to the other and just left outside, all of the base pairs disconnected. No, you need to open it segment by segment. See, if this segment is open, this segment and both of these segments remain closed because you do not want to damage these base pairs. They're very sensitive. So that's what you end up doing. And the other thing I told you, DNA polymerase, in this diagram where we were talking about DNA, where's the DNA polymerase diagram? So we were talking about DNA polymerase. I told you prokaryotes have polymerase 1 and 3, or 1 and 2, 1, 2, 3, all three of them. And eukaryotes have these symboled ones, alpha, gamma, epsilon, delta. So it's written Pol alpha and Pol sigma over here, which means this is DNA replication for a eukaryote, obviously. Okay, so that is one enzyme, DNA polymerase, done and dusted. Now the other enzymes are very, very easy. So I have one thing more that if you just to recall, you remember there was something we called a replication fork. It was exactly the point where the DNAs were separated for the replication into a fork, one twine of the fork and the other twine. Imagine this as these twines of the fork. So this is the replication fork. And what determines the replication fork is an enzyme called helicase. Helicase is an enzyme specially to break those hydrogen bonds. What helicase is going to do, look at this pointy one, it's going to come in, break these bonds where they're forming, and then open up the DNA segment, the specific segment that I need to replicate. It's going to open it up. Okay, so that is the purpose of helicase, and it doesn't work randomly. Let me just tell you something. Something that you need to know overall is that, so if this is your DNA, let's say this is the random DNA. It's obviously double-stranded, so it's a random double-stranded DNA. Now, helicase, for instance, what occurs in a eukaryote is that helicase comes in at this point, opens it, okay, and starts replication over here. At another point, it also does the same, opens it and starts replication over here. What that means is that in a eukaryote, replication points can be multiple. In prokaryotes, as compared to prokaryotes, where if this is the DNA, okay, it's circular, so it's closed, right? If this is the DNA, only at one point the helicase will come, open it, and lead to replication forming, right? It's not going to occur at multiple points. On a eukaryote, helicase can come in at multiple points, and no matter what point it comes in at, what matters is at the end of the day that it forms two different DNAs. Okay, so that's what really matters. Okay, where did I lost my slide. Okay, so that is one thing that you need to know. And if the question comes up, then we can discuss this further as well. But that is as much as you need to know for now. I don't want you to go into unnecessary details. So helicase is the enzyme that is going to split the hydrogen bonds open just to open the chain. Then once the chain is open, it can just fold upon itself. Hydrogen bonds are spontaneous. Then how do we avoid these bonds folding upon themselves? There are two specific enzymes, topoisomerase and single strand binding proteins. So single strand binding proteins are like dumbbells. What they do is they go and attach to this next to every nucleotide just to hold it open at the replication fork from both sides. You can see, look at these purple ones. They're holding it open. And then the other stabilizing enzyme is topoisomerase. It holds it, it's like a constrictor. Imagine it's constricting itself over here just to make sure that the area behind this does not become open, and the area after this point does not close upon itself. So it's obviously, it has an A's in the end, so it's an enzyme. These are just proteins, so both of them have just stabilizing function. So we are done with DNA polymerases and two stabilizing enzymes and helicase. The only thing left are these two enzymes, a ligase and a primase. Now, a DNA primase, what is a DNA primase? So DNA polymerase, like I told you, has two functions. DNA polymerase comes in and forms these bonds, right, to make this backbone. And it also excises a wrong nucleotide to form the correct bond, okay? These are the two functions of DNA polymerase: checking that the bonds are correct, that the base pairing is correct, and forming those bonds. But sadly, DNA polymerases cannot, only DNA polymerases cannot form the initial 3-4 bonds. Okay, it's very difficult for them to initiate DNA base pairing bond formation. They can form, for instance, if this is my strand, these DNA polymerase can come in and start working from here onwards, but these 3-4 bonds will be very difficult for it to form because of its enzymatic kinetics that you do not need to know about. So the body has adapted to a specific way for this. What happens is, now you know the difference between DNAs and RNAs. You remember ribose, deoxyribose, RNAs were uracil instead of thymine, and everything else was just same. Okay, so DNA and RNA nucleotides were just different by these two properties, if you remember. So now what happens is RNA, it turned out when we were studying these that RNAs are much more stable. The ribose forms of nucleotides are much more stable. So what happens initially is that in any strand in which DNA polymerase has to act, the initial few 3-4 nucleotides are RNAs, and those are called RNA primers. Okay, and primers, if you've done art or if you do makeup, you know what a primer is. It's used to prime a canvas, prime your face. It means just there to prepare it for what's going to happen. So RNA primers are, for instance, 3-4 nucleotides combined together. They're like a set of nucleotides. For instance, these three exist together already bonded. Now the DNA polymerase, obviously it's a DNA polymerase, so it can't work on the RNA polymerase itself, but it no longer has to initiate replication. All it has to do is an RNA primer comes over here, the RNA primer, and all of this happens during the S phase, remember? So the S phase gathers all these nucleotides into the cell, and it also gathers this RNA primer. So it attaches the RNA primer set over here, and then an enzyme called DNA primase comes in. DNA primase forms the first few bonds of the primer and the nucleotides and lets the DNA polymerase continue the chain. And at the end of the chain, DNA polymerase is going to go back. It's because it's a proofreader, so it goes in this direction, then it comes back and scans in this direction. It keeps scanning just to make sure it's excising the wrong nucleotides. When it comes around the second time, now the chain is complete, it's more stable. So what DNA polymerase at this point does is it removes those RNAs and replaces them by DNAs. Now it's very easy for it. DNA polymerase is a good exciser. It can easily excise wrong nucleotides. So it senses that it's the wrong nucleotide. It excises. See, because the covalent bond of this phosphate sugar is very strong when DNA polymerase makes it. So we cannot afford to have errors in it, which is why the nature has adapted in a way to put in primers and joined by DNA primase, and then finally DNA polymerase will replace those RNA primers with DNA primers. They were just 2 or 3, right? Not a lot of them. And on the leading strand is just probably the first segment only. On the lagging strand, relatively more, but then the DNA polymerase does the work, right? And then that's exactly what happens. That's how the whole chain is made. So this is what DNA primase is for the initial 10 nucleotides or 5 nucleotides that form the primer. And the last enzyme is something called a DNA ligase. And it's very simple. DNA ligase is like a stapler. It staples the Okazaki fragments or any other fragments or differences that are left in the DNA chain. So as simple as that. Even if you don't understand this, there's this table. You guys can screenshot this or screenshot it from the video if you want to watch the video later. So helicase unwinding of the helix. Gyrase is the same as this topoisomerase, same as this one. It's a stabilizing one, assists in the unwinding and stabilizes the DNA. Small single segment binding proteins, these ones, okay, they're removed from this diagram, but in the previous diagram you could see them. They stabilize again. Primase synthesizes the RNA primer, so that the primer can come in and attach, and then DNA can start its working. Okay, and then DNA, both of them have been told here, the polymerase 3 and 1, which are prokaryotic. So polymerase 3 elongates the chain. 1 is going to remove and replace the primer, just like I told you, and fill in the gap with DNA. So essentially, the overall working of polymerase is both elongation, excision of wrong nucleotides, and removal of primer and its replacement. So 3 important functions. And then the DNA ligase, which closes the phosphodiester gaps given here between the Okazaki fragments or any other gaps that may exist. Okay, and this is a continuous process, continuously happening. Again, you can see the helicase breaking the bonds here, the single strand binding proteins stabilizing the strand. You can appreciate this primase. It has added a short primer template, and this is not a DNA template. I just told you it's an RNA template, a much more stable template, but a template that is relatively easier to remove as compared to DNAs that, if become wrong, cannot be easily removed. Okay, then the polymerase, which has 3 functions. And then this is one of these two are both polymerases. And then finally the ligase to join the Okazaki fragments, to join any nicks in the backbone that we have. So that is DNA replication for us. You understand DNA polymerase, you understand the leading and lagging strand, the ligase enzyme, and you also know what a gene is. In case anybody had missed my topic 1, which I think a lot of you guys have. So what in topic 1, I told the class what a gene was. And just to repeat it for you guys, a gene is a segment on a chromosome that codes for a specific protein. And the Amoeba Sisters video that I sent you, it explained the same thing to you. There's a segment of the DNA that goes under transcription and then translation to give a protein. That segment that is a coding segment, or that codes for a protein, is called a gene. If it does not code for a protein, for instance, if you remember telomeres did not code for proteins, then they're not genes. They're something else. They're non-coding, okay? And then finally, we have that special term called semi-conservative replication. So semi-conservative replication, so whatever we've studied up till now is semi-conservative replication. But at this point, if I ask you guys to tell me that, do you understand what semi-conservative replication means? I'm sure most of you guys do not. Do you guys understand what semi-conservative replication means? So tell me.
The semi-conservative, okay, I'll try to explain it, but I'm not sure if this is what it means.
Ahead.
A semi-conservative means that it contains one strand from the which is like a new form strand, and the other strand is the original strand. So there are two of those kinds, I think.
Okay, yeah, sure. That is a good definition. That's true. Anybody else was trying to say something?
I don't fully understood it, but I know the basic idea of it.
Okay, so yeah, which is why I had to add a few more slides into my presentation, because I realized that when we keep talking about semi-conservative replication, most people don't really understand it. But yes, Tanya's concept is correct, and that is exactly what we've been studying here, right? An old chain and a new chain forming together. But now we need to understand semi-conservative replication from the point of view of a scientist, which is why we need to know what actually is happening in semi-conservative replication. So there are essentially, when scientists thought of replication of the DNA, they thought of three different ways. They thought of a way that is conservative, another one which is semi-conservative, and then a dispersive way. And then there are many other ways, but these are the simplistic ones, and these are the ones that you might get tested on. Now, you need to understand what a conservative way is versus a semi-conservative versus a dispersive one to reach the conclusion that you have what is a semi-conservative replication in your cells, okay? So a conservative one simply means if I make up chains, made these blue chains. Okay, so before moving on, we need to talk. Okay, so there's one thing that we didn't do when we were talking about ribosomes initially, is that you guys know, and you guys told me that there's something called 70S and there's something called 80S, eukaryotic and prokaryotic, right? You remember that, but do you guys even know what 70S or 80S means?
Yes.
Okay, what does it mean?
The subunits of the ribosomes, the bigger unit is 80S and the smaller subunit of the ribosome is 70S.
No, that's not exactly true, because this 70S itself is made up of something called a 40S, something called a 30S and a 50S. And this 80S is made up of something called a 40S and a 60S. So then the subunits, the larger subunit is the 60S and the smaller is the 40S, and the combination is an 80S. So your definition is not slightly, but it's quite incorrect. This isn't something you're taught in your A-level a lot because it's not relevant, but when you're doing, for instance, medicine or your normal biology courses in your university, that's when you get to know this. But I want you to know it right now because it isn't that complicated, because it is something that's going to help you with the concept of ultracentrifugation. And you guys do have the concept of ultracentrifugation in your A-level. So you're supposed to know what centrifugation is, right? So this S given right here is called the Svedberg unit. Svedberg was the scientist who found out these ribosomes. What he did was he took a test tube and he lysed eukaryotic cells and put them in, and prokaryotic cells and put them in. And when he centrifuged the cells in this plasma, what he realized was that there was one settlement or sedimentation at this level, and then another sedimentation at this level. And so whenever he would lyse eukaryotes, the sedimentation would occur at this level, and at prokaryotes, it would occur at this level. When you ultrafugate something, the sediments always settle to the bottom. This Svedberg unit is in fact what indicates at the location where these sediments settle. So obviously, if you know the basics of centrifugation from O-level, you know that the heavier substances settle at the bottom. So there was eukaryotes had ribosomes that always settled at the bottom. So he named them 80 Svedberg. 80 is the unit for this location of settlement on the test tube. Like, you know, like those volume beakers in your chemistry labs that have units, 20 centimeter cube, etc., etc. The same way, 80 Svedberg is for this ultracentrifugation. 70 was for this one. Now, why is this so? If this is clear, then we can move on. Is this clear?
Yes.
Do you guys understand what the Schwetzberg units are? If you want to read more about them, go Google them. But I don't think you need more information on it. That's as much as you need to know. Okay. So why did you need to know this? Because there is this one experiment in A-level that started coming up in your papers a while ago, and it's important that students know how to go about it. So what's happening here is, so essentially we had three theories: that DNA is conservative, which means if I put in a strand of my eukaryotic cell, I lyse it, put it in a test tube, and add free red colored nucleotides to it, then the nucleotides will use this strand, both of these strands, to form individual strands, and eventually both of these DNAs will be totally separate, which means all of these red nucleotides form a separate DNA. They've used the template of this one. Both of these are identical, but they form separate DNAs. They do not mix with each other. And how did I assume that was happening? I tagged these blue ones. I put a weight of 10. For instance, I put them on the weight of 10, and the red ones I made on the weight of 5, right? So the heavier 10 ones settled over here to give me this blue line, and the lighter red one settled over here to give me this 10 line. What that meant was that after all the replication took place, when I added my lighter nucleotides to this heavier chain, both of these chains were no doubt identical because nucleotides used this copy to create their own. But this chain was completely itself and heavier, and the newer chain was only made up of the nucleotides that were lighter and that I had put in. So it gave me two sedimentation areas, as compared to a semi-conservative replication, where when I put in both of these, when I initially took the test tube, it only gave me the reading of a blue line. But when I put in the lighter red ones, what happened was when I ultracentrifuged it, it gave me one single line at in the middle of 10 and 5. It gave me a line in the middle, exactly in the center, which meant that every DNA in this test tube, all of the DNAs had half retained themselves, half of them were 10, and the other part that they had replicated was in fact made up of 5 nucleotides, and they were also identical, but half of each DNA was the original one and half of it was of the weight of 5, which is why its total weight came in the middle, and all of them gave me a similar weight, as compared to dispersive, where I got a dispersion of weights across the test tube, which means some of the DNAs might have, let's say, 70% of the heavier weight and 30% of lighter weight. Other DNAs were 90% of the heavier weight and 10% of the lighter weight. And even if they were identical, they had different weights, which meant that the nucleotides had dispersed between each of the DNAs. So what were the three DNAs? Conservative, where the new nucleotides had formed, all three of them were identical chains, new chains, but conservative had a separate DNA. Semi-conservative had DNA that was half here and half there. Dispersive was mixed in. It could be anything, which is why you get many readings, many dispersions across the range. You get many dispersions. So is this clear?
Yes, miss.
Yes, Isha.
Yes.
So, Rima, do you understand what it means when we say semi-conservative as compared to any other way?
Yes.
Okay, and so even if you don't, then this is another example. As you can see, dispersive means any arrangement can take place. Conservative means the old one remains itself and the new one is added that is totally separate. Semi-conservative means half of the chain is new and half of it is old. When we put in these heavy and light chains in real life experiments, what we realized was that in eukaryotes and prokaryotes, even this one is cut out. The only reaction that is taking place is semi-conservative. Okay, the only DNA replication that is taking place is semi-conservative. And again, this is the illustration for semi-conservative. So I hope this is clear. Okay, So now we have how much time do we have left? So we have 10 minutes left, and let's see how much we can do, and the rest we'll cover tomorrow. So the next thing that's left for us is the protein synthesis. Protein synthesis has a number of steps. There's a number of more definitions that we will hopefully do, and you guys will understand. Is the DNA replication and everything we've done up till now very clear?
Yes, Miss.
Will you guys now be able to answer the questions?
Yes, teacher.
Okay.
Yes.
That's great. Good to know. Okay, now we need to know protein synthesis, and it's going to be very easy if you watch that video. The only thing I'll have to clear is the little bit of confusions. So if you watch that video, if there's any confusions while I'm explaining, just tell me right there and I'll address those. So the rules are simple. We know those rules. DNAs make RNAs and RNAs make proteins. Before we move on over here, I'd like to explain one more thing that books and teachers won't explain to you, but I think are essential for understanding. RNAs exist in 3 forms. And you've heard of them, but you just can't characterize them yet. There's something called an mRNA, there's something called a tRNA, and there's something called an rRNA. And all of these 3 work together to give you this protein. So just remember, they're working together in coordination to give you this protein. mRNA, and all 3 of them being made in the nucleus in one part or another. For instance, if you remember the nucleus, there was something called the nucleolus. Remember? That nucleolus is what makes the rRNA. And where do we get the mRNA from? We get the mRNA from this genetic material, from the coding DNA, and we will see why. If you've watched the videos, you already know why. But even if you don't, just know here that tRNA comes from all of these mixed sources from inside the nucleus. Okay, so rRNA from the nucleolus, tRNA again from inside the nucleus, from another structure that is very specialized and beyond your understanding, and then finally mRNA. All 3 of them have separate tasks to perform. mRNAs are used for, because they are a copy, they're made from the coding gene, they are a copy of the message of the gene. And the copy, and they're made of obviously, why are they called RNA? Because they're made up of the nucleotides, right? And only the ribose type. And like I told you, ribose type is more stable. So what happens is DNA, we don't want to damage it because it's the final code. It remains inside the nucleus. Then we know that protein synthesis, we've been saying, is occurring on the rough endoplasmic reticulum. Then something needs to come outside from the DNA over here for the protein to form and go to get modified, right? And that something is the mRNA. It's a copy of the coding gene of the DNA. It goes outside towards the rough endoplasmic reticulum to give you the information of what should be made. For instance, one person has a gene for tall, right? Another person has a gene for short. Now, this person's mRNA is for tall, this one's is for person A and person B, right? For instance, Aruba and Abdullah, I don't know who's taller, but I'm guessing Abdullah is taller. So
Yeah.
so Abdullah has the gene for tall, and Aruba has the gene for short. But both of you have been eating the same food. Both of you were born to the same parents. So how does your protein of your muscular protein, your DNA, tell your muscles and bones to grow taller, and Aruba's doesn't, right? That is all in control of this mRNA. And how does the mRNA control it? So we'll talk about that. But until then, let's talk about these two, tRNAs and rRNAs. Now, once I've told you the function of mRNA, which is essentially bringing in the code for protein synthesis, right? It brings all of them form protein, like I told you. This one brings in the code. tRNA brings in the amino acid. Okay, remember this. It carries the amino acid. Now, both of you will have the same tRNA, same amino acids, because you eat the same food, you live in the same place, same amino acids are being carried in every part of your cell, then how are they responding differently? Because at the end of the day, when the proteins are being formed, mRNA and tRNA are interacting together. Their addition is deciding, the mRNA is helping the tRNA. tRNA will bring all kinds of amino acids that are same, but the mRNA, for Abdullah, will decide that only these certain amino acids will arrange together to form certain proteins, right? So it's going to select, the mRNA will dictate the tRNA to bring certain amino acids to form certain proteins for growing taller. So that's essentially all that happens. And then finally, we have the last component, the rRNA. This is the component that forms those ribosomes, the ribosomes that we just talked about, the Swedsberg ribosomes, okay, the ones that we just ultra centrifuged. So this is, the lecture is going to be over. Rejoin the same one, okay? Whenever it's over, just make sure you rejoin. Okay.
Okay.
So that is now our RNA discussion. Now let's move on from the RNA discussion to the next part of the discussion, which is this. This diagram is beautiful in its explanation. See, it's showing replication of the DNA over here. And then it's showing two pathways, replication over here, and over here it's showing the transition from DNA to protein. How there's the first step of forming transcription to give us an RNA. And then that RNA is translated, or translation takes place to give us the protein. So these are essentially the only two